Good afternoon and welcome. I'm Chris Fanta. It's my pleasure to welcome you to Asthma Grand Rounds today, including those of you who are joining us by live webcasting. And our theme today is that always challenging problem of severe asthma. And I'm pleased to introduce as our speaker on the topic Dr. Marcella Ruddy. Dr. Ruddy was a resident in medicine at Beth Israel Deaconess Hospital and then came and did her pulmonary and critical care fellowship here at the Brigham and Women's Hospital. And she joined the faculty in pulmonary and critical care medicine at the Mass General Hospital, where she headed the adult CF program. And then went off to work at Merck, where she was clinical lead for their RNAi program, and ultimately came to lead the immunology early clinical development program there, and spent about 9 years at Merck, and then went to EMD Serono, where she was vice president for clinical immunology. And just now in the last year, she's joined Alnylam, where she's vice president for drug safety and pharmacovigilance. And while at Merck, Dr. Ruddy helped arrange a series of presentations by Partners Asthma Center faculty, where we had the chance to come over to Merck Labs and talk to her group, the basic science group, about what Partners Asthma Center was doing and what our staff were doing around asthma. And I always thought, wouldn't it be nice to get this as a two-way conversation, to hear a little bit about what you and your group was doing around asthma. And so here's the opportunity for payback. I'm very excited to introduce Dr. Ruddy to talk about the search for biomarkers in severe asthma, perspective from Pharma. Dr. Ruddy.
Hey, Chris. Thanks very much for that introduction. It's terrific to be here today, and I'm really happy to see some familiar faces in the audience. I'll just start with my disclosures and disclaimers here. As Chris said, I am a former employee of Merck Research Labs, which is where I have done the bulk of my drug development work. And just wanted to emphasize to everyone that that the views expressed in this presentation are really just mine and not representative necessarily of any past or previous employer. And very intentionally, I want to point out that I don't plan to speak about any marketed Merck products in this presentation. And I do refer to a drug candidate in this talk as part of an example of how we're leveraging new biomarker platforms for drug development. And in that setting, I'll just talk about drug X. So what I hope to accomplish in the next 45 minutes or so is to talk a little bit about what the world of drug development is like and what the problems are that we face, and try to explain to you and discuss with you why it's been so difficult to develop new therapies for severe asthma. I think, as all of you know who treat asthma patients, our productivity in the drug development side for new therapies for asthma has been somewhat thin over the last, say, 15 years or so. And I wanted to help kind of put into context why that might have been the case, and why perhaps, and hopefully, a better understanding of the underlying biology of severe asthma and really using more targeted biomarker approaches may hope to help improve the probability of success. And here I hope then to present a few case examples of how the use of biomarker in drug development may help to improve our successes by using a few examples of drugs currently in development. So the challenges in drug development are large in that it costs an awful lot to develop a new drug. Over $1 billion is spent in research and development to get a new drug to the market. And unfortunately, there's a very low success rate in this endeavor. For over 1,000 compounds that might enter preclinical testing, only 10 will make it into clinical trials. And of those, only 1 will eventually get FDA approval. And combined with that, unfortunately, there's still a lot of late stage failures, those products in phase 3, where a bulk of money has already been invested in those development candidates. Over a third of those are still failing in phase 3 drug trials. And all of that rolls up to actually leading to a fairly poor return on investment in drug development, where only 2 out of 10 approved drugs bring in enough revenue to recoup their development costs. The problem of probability of success in drug development was recently updated and highlighted in an article in January of 2014, where they really analyzed 835 companies and looked over the period from 2003 to 2011 on what the success rate had been in drug development in phase 2 and phase 3. Here on the left you see a chart in which it looks at probability of success across a range of therapeutic areas for drug targets. And you can see in phase 2, the failure rate is ranging between 20 and 30% success rate in phase 2. And again, that's not all that surprising, because that's the phase where you really begin to test your drug in the disease population to assess for efficacy. What's particularly disheartening, however, is that you're still seeing a significant amount of failure in the phase 3, where you have across these therapeutic areas still success rates that are in the 60 and 70% rate, where even in oncology, I'll highlight that there's a greater than 50% chance that you could still fail in phase 3. This all rolls up to the likelihood of an approval for any drug entering phase 1 of clinical development ranges from 18% down to 7% for new clinical entities. You can see here in the respiratory disease category, we're sort of at the mid-range of about 11% likelihood of approval. Now, the reasons for failure in clinical trials is outlined here, and again, not so surprisingly, often fail due to efficacy, with safety being another primary reason for failure. So you can really simplify the reasons retrospectively and somewhat painfully for why drug candidates fail. It tends to be because you've chosen the wrong target, the wrong molecule, the wrong endpoint, or the wrong patient population. For severe asthma, this has been particularly a problem in terms of trying to select the right target. The wrong target has been selected many times, you'd say, because really the underlying pathobiology of asthma has not been well understood. It's generally a clinical diagnosis. And so the heterogeneity of particularly the severe asthma spectrum has been unappreciated in terms of trying to develop new targets and try to pick populations for drug development. The wrong molecule, that's a common problem in drug development and is not particularly unique to severe asthma. But particularly, and as we'll discuss later in the talk, some issues about selecting the right endpoint for drug development of novel compounds, particularly targeting severe asthma, and also selecting the right patient population have been particular stumbling blocks for severe asthma. For the endpoint, FEV1, an excellent surrogate endpoint for targeting mild, moderate asthma. There, that population, as we'll discuss later, tends to be a generally atopic patient population that's very responsive to bronchodilators and inhaled steroids. Whereas the severe asthma population is more complex, where really endpoints of interest for those patients are really, can you improve exacerbation rates, symptoms, quality of life, and changes incrementally in FEV1 don't really play an important role. Additionally, understanding the right patient population amongst the heterogeneity of asthma has led to an inability to really get success in an all-comer severe asthma patient population. So how can we improve this process? One way to go about that is at least understand what your drug is doing in the early development space to make sure it's an adequate drug to test in late-stage clinical development. I've spent most of my time in pharma really thinking about biomarkers and platforms for really understanding this early development space and how do we really assess novel compounds to make sure that we really understand the underlying biology and pathways that we're trying to impact, and make sure that these novel compounds at least do what we think they're doing in the body Do they hit the target that we have designed this molecule to hit? Can we measure that appropriately and accurately in humans, like we did in the preclinical animal species? And can we then demonstrate that hitting that target actually alters the biology and confirms the hypothesis that we think of why this pathway and this novel mechanism that blocks that pathway will have relevance? So in early development, we really develop, we try to develop biomarkers that'll actually help us understand that. And these are useful biomarkers for understanding safety, toxicity sometimes, understanding whether you've hit the target, understanding why you've impacted the biology. But importantly, these types of biomarkers and these approaches may not necessarily inform on the actual eventual efficacy of a drug in a phase 2 or phase 3 trial. It really tells you that the drug is hitting the mechanism, hitting the pathway, but then the true test of whether it's actually going to work in asthma and in severe asthma has still been a huge stumbling block for development of novel asthma therapies, because we don't really have any good biomarkers to predict efficacy in an exacerbation endpoint. So, but at least if you can hit these, at least if you can understand where you've hit, where you've impacted the pathobiology, that at least helps you understand whether your molecule is worthy of going into phase 2 development. So I've spent a lot of time thinking about how we can innovate and improve that process. A classic way of evaluating new drugs has been the allergen challenge, and that's not a new platform. Many drugs, in particular inhaled steroids, have demonstrated quite a useful benefit in this inhaled allergen challenge. Many of you may know what this model is. It generally, what it does is it leverages mild asthmatics that are known to have an allergic component of their disease. They're given a titrated dose of allergen, and this induces a predictable inflammatory allergic response in the airway. This response typically has a dual FEV1 response, an early response that occurs within the first few hours of exposure that is a result of allergen binding IgE and activating mast cells and releasing mediators that cause an acute bronchoconstriction and a decrease in FEV1. That's then followed by a late response within 3 to 8 hours post-allergen, where you see the cellular infiltration and edema that then causes a second dip in your FEV1. Over here on the right, you can see an example of an allergen challenge that we conducted at Merck, but is very comparable to many allergen challenges that have been done, where you can see in blue the effect on an FEV1 with a placebo treatment, both in the early and the late phase. And then you can see the attenuation of this reaction after, actually in this study, a very low, very limited number of ICS doses, 5 doses over 2 days, was actually able to completely attenuate in this study the late stage drop in FEV1 and actually impacted the early phase drop significantly as well. Now, this has been useful in terms of a negative predictive value for drugs in development, in terms of drugs that haven't demonstrated a significant improvement in LAR response have generally tended not to then go on to be efficacious in asthma, chronic asthma. But the positive predictive value hasn't been terrific. There are definitely drugs that have shown an improvement in LAR by FEV1 and then have gone on to fail in clinical asthma studies. So it's not a perfect assay, but what we, in my group, were thinking about is what can we leverage, what can we learn about what's going on in the airway on a molecular or cellular basis that might help inform drug responses. And so what we did was to look at whether or not the impact of allergen on the sputum inflammatory response could be a useful way to interrogate the pathway of our drug and its impact. So we went about trying to optimize sputum RNA collection, and then we did gene profiling on sputum, looking at individual RNA effects on individual genes, looking at effects on immune cell infiltration and changes in gene fraction as a reflection of gene signatures, and then looking to see if we could actually see impact on pathways and cell activation by leveraging RNA responses in the sputum. So the way these slides are set up, I'll just show you. They're set up to look at change from baseline in RNA expression. And so by definition, baseline is at 0. The placebo arms of the study, those that didn't get any pretreatment with drug, that are allergic asthmatics exposed to allergen, have then, if they have an effect on a gene signature, will have a big allergen effect. And if the drug attenuates that, you'll see the change in that placebo effect from treatment to drug, and that'll be the drug effect. So as an example, here I'll show you what we found with looking at just the IL-13 gene expression in our allergen challenge model and compare it to what we saw with fluticasone versus drug X. And importantly, we also wanted to see effects over time after allergen exposure. So we collected sputum samples at both sort of the peak of an LAR, around 7 to 9 hours post-allergen exposure, and then again at 24 hours post-allergen. And as you can see here on the right, the placebo arm has a big allergen exposure impact on expression of IL-13, as you would expect, and fluticasone essentially quenches that response. And again, you can see still some effect of allergen at 24 hours post-dose on IL-13, and again, you see fluticasone is able to attenuate that response. In evaluating what our drug would do, which is an oral controller that was targeted against inflammation, Th2-like inflammation, we can see that it is having some effect on IL-13, which again confirms what we thought the biology of this molecule should be. Now you can look at this across the spectrum of Th2 cytokines, and again we're seeing similar patterns with fluticasone versus our drug for IL-4 gene expression, IL-5, and IL-9. You can then also ask the question, if you have an understanding of the biology of your drug and you want to see if it has a unique effect on a gene signature, you can look very specifically. And here we just to check our system, we chose to look at a classic transactivating gene that's influenced by glucocorticoid exposure, FKBP5. And here in this example, not unexpectedly, it doesn't have much impact with exposure to allergen, but this gene does seem to be increased in expression after exposure to glucocorticoids and doesn't have any effect with our drug. Again, we wouldn't have predicted it would be the case, but it did confirm that our system does seem to be manifesting some of the predicted responses that we would see. We then look to say, can we use this approach to actually maybe improve looking at cell populations within sputum? Many of you have done allergen challenge studies and looked to count cells from sputum. You know it's technically possible, but introduces a level of complexity in clinical trials. And so we wondered if we could use an eosinophil gene signature and how well that would correlate with sputum's eosinophils that are assessed by a routine cell count. And here you can see that the correlation between sputum's counted in cells versus utilizing our eosinophil gene signature, there was a really nice correlation between those. And then we assessed sort of the technical success of looking at these two, and we found that, not unsurprisingly, we lost a lot of samples for our eosinophil cell count just because they weren't adequate for processing, and we had a better yield by looking at our eosinophil marker. So this suggested to us, just as a tool perhaps in drug development or in clinical trials, this might be a useful approach. Then we looked at whether we could leverage this even more to understand pathway biology of the impact on different gene pathways by our drug. And our informatics group looked at this gene set across 4 or 5 allergen challenge studies that we conducted and did a cluster analysis and was able to define clusters of genes that change together in response to therapies. And here, again, just as a demonstration of how this might work, a gene cluster list was generated. And again, here we're seeing fluticasone, allergen having an effect on a Th2-associated gene cluster and being attenuated by fluticasone, and our drug having somewhat of a similar effect on that gene cluster. And then you could again see clusters of genes that might be impacted by, in a differential way, by fluticasone versus our drug. Here is an example of a CD8 positive gene profile that our drug, that the placebo in an allergen wasn't greatly affected by. The gene signature by allergen wasn't greatly affected, but fluticasone did impact that signature, and our drug did not. So this begins to sort of show you in a way that this kind of approach with allergen, or really with almost any airway challenge model that you might want to test, it might provide a method where you could interrogate your drug in an early development space to really better appreciate how you're impacting inflammatory pathways and how you could maybe discriminate dose effects so that you can dose focus for selecting what the right dose range and going into your later stage clinical development programs. Obviously, this is a very limited type of platform. It's not going to be predictive of success in late stage, but it gives you a handle on your impact on biology. And also, notably, it captures inflammatory cells in sputum, so that if your target of interest is predominantly in your epithelium or smooth muscle, this might not be a useful platform. So those are ways that we've really tried to impact early development. But the big hurdle in severe asthma, many would probably argue, is how do we improve late-stage severe asthma drug development? What can we do? What biomarkers? How can we understand better and predict disease courses in these asthma patients to be able to better find biomarkers that could define molecular changes that are associated with disease progression and help us select patients to improve treatment outcomes. And importantly, help us improve the benefit risk. Any drug has risks associated with benefits. And if we could predict by biomarkers those patients that are more likely to respond or have, on the converse side, worse side effects, those kinds of biomarkers are very useful and needed in severe asthma. So going forward, what progress have we made? And there has been progress to try to tease out and try to help find some of these biomarkers. And some of this work has been done by many of you working in asthma research, by the consortia of severe asthma physicians and scientists, and trying to really at least start to understand the different phenotypes that make up the severe asthma diagnosis. And this has been done by biased phenotyping and unbiased cluster analysis. And over the last several years, there have been at least 2 large unbiased cluster analysis studies that have helped us understand a little better some of the characteristics to help define the heterogeneity in asthma. One classic study by Halder, which represents the asthma network in Europe, showed that in 370 asthmatics, they were able to actually categorize severe asthma by sputum cell counts and symptoms. And Wendy Moore and the SARP group in categorizing and studying the severe asthma population here in SARP was actually able to define 5 separate clusters, with the strongest predictors being FEV1 and age of onset. This is probably a slide that's common to many of you that depicts the defining of these clusters that's predominantly done by FEV1 and age of onset. And looking at these clusters, it was appreciated that there were certain characteristics that were common to some clusters and not to others. And in this, it was found that clusters 1, 2, and 4 were more involved, more patients in those clusters tended to be atopic, tended to have eosinophilia, tended to have more responsiveness to inhaled steroids. And in clusters 3 and 5, they tended to be not necessarily atopic and have more of a neutrophilic inflammation in their airways. And this has led to the beginning of thinking about and dichotomizing asthma somewhat to being Th2 dominant and non-Th2 dominant. Now, obviously, this is highly simplified, but it helps to kind of begin to think about how you might target new therapies. And so I'm going to talk a little bit now about 2 development programs that were really focused in this area of atopic asthma, eosinophilic asthma, and sort of the Th2 dominant asthma. And talk about these 2 programs that I selected are the meprolizumab development program and the lebrikizumab development program. Meprolizumab is an anti-IL-5 antibody, and lebrikizumab is an anti-IL-13 antibody. And I'm going to compare and contrast the approach that was taken in these development programs and suggest that having a better understanding of the heterogeneity of asthma, I think, would have impacted these programs in a way that would help expedite the drug development process. So meprolizumab, this is a paper that was published in 2000 on the anti-IL-5 antibody. The hypothesis for IL-5 is it controls trafficking of eosinophils. And so the hypothesis had been, and it was that eosinophilic inflammation is fundamental to asthma pathogenesis, and that by inhibiting IL-5, you could inhibit neutrophilic inflammation, and that would then lead to an improvement in airway hyperreactivity, FEV1, and asthma symptoms. And preclinical models it definitely predicted that that would be the case. And then in a clinical trial that was done in 24 asthmatic subjects, they definitely showed impressive improvements in blood eosinophil levels in 2 different doses of IL-5 antibody. They showed an improvement in sputum eosinophils with a suggestion of a dose response. But they importantly didn't show any improvement in histamine PC20 or an assessment of airway hyperreactivity. Because, however, this actually hit its biomarker-driven hypothesis of inhibiting eosinophils, so this then went to phase 2 clinical studies. It went to phase 2 clinical studies, however, in all-comer asthma. The concept was that all patients with asthma have eosinophilia, and thereby, if we block eosinophils, we should have effects in asthma. And importantly here, they picked the endpoint of FEV1. So with this sort of all-comer approach, with a 12-week study looking at lung function endpoints, you could see that it absolutely had effect on blood eosinophilia. But here in this graph here, looking at effects on FEV1, you can see that there really was no significant difference in FEV1 in the all-comer asthmatics versus placebo. So, although this failure, I'm sure, must have been disappointing, those that were studying this drug did not give up. And this was probably concurrent with a lot of evolution of understanding of heterogeneity of asthma and led people to wonder, is there a subpopulation of severe asthma for which this drug might be potentially useful? So, in 2009, there was a study published that looked very specifically at patients with high eosinophilia that were resistant to steroids. In here, they chose a new endpoint. They chose reduction in exacerbation. So it was a longer study. And they looked very specifically in patients that had sputum eosinophilia of greater than 3%. And in this case, on the endpoint of exacerbations, they were able to show a 40% reduction in asthma exacerbations in this patient population. Importantly, they were unable to show any improvement in FEV1 or airway hyperreactivity or symptoms. This then led to phase 3 studies, which were published in the New England Journal in 2014, were much larger clinical studies with, again, a long clinical study course of 32 weeks. They were able to see, again, with 2 different doses of mepolizumab, an improvement in exacerbations over placebo. And again, they showed a clinically insignificant change in FEV1. So essentially, this drug has perhaps found its niche in that high eosinophilic asthmatics may well benefit from this drug. But if you'll note, the first study that I showed you was in 2000, and here it is, 2014. So it took 14 years of sorting this out to be able to hopefully get a novel drug on the market for asthma. So here then I'd like to contrast slightly with you the development of lebrikizumab, an anti-IL-13 antibody for the development of severe asthma. Now this was staggered in time and most likely benefited from the experience with the anti-IL-5 drugs, but was done in a way that really seems to be biomarker driven. They have collaborated quite closely with Genentech has collaborated quite closely, it appears, with UCSF and asthma researchers in that institution. John Fahey, Prescott Woodruff have been involved also in exploring the hypotheses in other asthma cohorts and looking at what's the level of, is there a differential in IL-13 expression in certain asthmatics versus others? And could that be a differentiating feature of a certain subset of severe asthma? And in this study that was published in 2009, Prescott Woodruff demonstrated that amongst asthmatics, he could discriminate asthmatics based on a gene signature of IL-13 responsive genes. And in this gene cluster of periostin, CLICA A, and serapin B, he could discriminate 2 different asthmatic populations. And this asthma population also demonstrated, when measured, high levels of IL-13 in bronchial biopsies and was also associated with high levels of BAL eosinophils. On histology, there was also a difference in these patient populations in that IL-13 high patients tended to have an increased level of eosinophilic inflammation. They also had evidence of increased subepithelial fibrosis and an increase in mucus composition, suggesting that this is a slightly different phenotype than may have been previously appreciated. And using this approach, it was noted that periostin is actually a secreted measurable protein. And so the hypothesis was put out in the development program that those patients that have higher circulating levels of IL-13, of periostin, are likely to have high IL-13 tone. And so those might be the patients that might benefit most from an IL-13 directed therapy for their asthma. So essentially in their phase 2b study, they took an all-comer severe asthma approach, but they had a pre-specified analysis to look at high periostin versus low periostin asthmatics. And here you can see they were able to see some change in FEV1 in the total cohort over a period of 12 weeks. And that all-comer difference in FEV1 was, marginal. However, if they separated out the patients from a high periostin versus low, they did begin to see a differentiation in their FEV1 responses, suggesting and confirming the hypothesis that those patients with high IL-13 tone might respond more to an IL-13 agent. So as those results came out, we have seen on clinicaltrials.gov that they have launched quite extensive phase 3 studies for this compound in duplicate. They're looking to see in what appears to be a general all-comers asthma population, but at a very large number for an asthma study, over 1,000 patients. They're looking at 2 doses versus placebo, and they're looking at improvement in exacerbation rates after 52 weeks. What I think is likely going on here is, of course, in trying to prove that they have defined their selected patient population, they're also likely looking to demonstrate that they have an assay for periostin that is able to discriminate and identify this patient population, and that the assay is then suitable as a companion diagnostic for when this drug, hopefully for this company, demonstrates benefit. It will then come along with a companion diagnostic that will help clinicians understand whether this drug will be useful for patients. As any of you that are following this, which many of you likely are, if these drugs are successful, a wave of these things is going to be coming towards you. And in these Th2 flavored therapies for severe asthma, and many of them are overlapping, and this Venn diagram is just meant to suggest that they overlap. It's not meant to in any way suggest one is greater than the other, but you have a whole host of compounds. I highlighted lebrikizumab, but just as an example, there are a number of Th2-driven agents that are in development. There are a number of second-generation anti-IgE therapies that are in development, and then there are a number of anti-IL-5s in development. And how, as a clinician, are you going to be able to assess which of these is right for my patient, which is going to be best? And actually, it's going to be on the onus of the company to help you understand that and provide the right diagnostic tools to help you understand which drug is best for your patient. I haven't talked a lot about treatment for non-Th2 high asthma. That's obviously another group of diseases. It's likely to be also very heterogeneous. And so the idea that you just go for non-Th2 asthma and you've got your target is likely too simplistic. It is heterogeneous, although there is some evidence that the neutrophilic inflammation there may be amenable to drugs that impact neutrophils. A recent trial was done with brodalumab, which is an anti-IL-17 receptor antagonist, which also notably hits IL-25. However, in an all-comer asthmatic study in steroid-resistant asthma was demonstrated to have no obvious clinical efficacy. Subset analysis did suggest perhaps a population with high degree of reactive airways that might benefit. And there is now a second trial ongoing to evaluate that question. But notably, they don't seem to have any biomarker that can identify patients with high IL-17 tone. And so it's unclear how that lack of a direct biomarker will impact that program. So I hope over the course of this talk I've helped show you how biomarkers can be phase-specific and have specific questions that they try to answer, all along the way trying to build the weight of evidence that a compound is having intended biologic effect, and that biologic effect will impact disease. As you go through development, you need to increase your understanding of these biomarkers and how they can then help clinicians, help all of us understand which patients will benefit, and therefore what's the rationale for the value of this new therapy. Although I started the talk speaking about all the challenges in drug development, what I have to say is that the opportunities now way outweigh the challenges, I think, in terms of it is a great time to be in medicine and to be in drug development. The scientific advances that are occurring now, both in platforms and understanding of biology, the technologies and the ability to develop new drugs are rapidly improving. Our understanding of big data and how we can actually incorporate features of phenotypic data with genetic data and really try to understand diseases in a deeper and more scientific way will help us design drugs and design biomarkers for drug development. But importantly, I've also hoped to emphasize in this talk how important it is that we all collaborate, that I don't think we can be successful if pharma is siloed from clinicians and from academic researchers in asthma and in all diseases. I think we can only be successful if we can really enhance collaboration. So I'd like to acknowledge my collaborators at Merck for all the work they did on some of the work that I presented, and just say thank you for listening.
Thank you very much. That was beautifully done.
Oh, you're
Thank
welcome.
you. Stay right here, and
stay right here.
like to take some questions if you have them. I'm curious, I just get us started. Do you remember, among a population of severe asthmatics, what percentage is likely to have high periostin phenotype? Is it a small percentage? Is it a large percentage? I don't remember the study, but...
yeah, I would guess that it's a very small percentage, along the lines of what has been seen with the high eosinophil, which is around a 3%. But Elliot is probably the expert here, so I'll defer to him.
Remember the percentage in the study?
They didn't really study, for the lebrikizumab study, they didn't really study severe asthma. It was kind of moderate to severe. It wasn't these people who were on high-dose steroids or whatever. So they actually took mean periostin level and just said high, mean, low, mean. So they had 50-50. But I think you're right. As you get into the severe asthmatics, that's a smaller population. And the interesting thing was, if you look in the supplement in that study, nitric oxide was as good as periostin, or better actually, than periostin in terms of identifying the people who were going to respond to lebrikizumab. And we know that the nitric oxide levels in the patients with the high steroids are maybe 15-20%, in the patients. So.
Are you able to tell us what the eosinophil gene signature was that corresponded in the sputum eosinophil counts?
Sure. The question is, can we talk about the gene Th2 signature that was seen in the sputum? That's an excellent question. I believe that there's a publication being prepared, and so hopefully we can share that in that publication.
And this might be related to the same question. I was wondering if in the eosinophil gene signature you found, Fahy and Prescott found the three, right? Periostin and serpin and I always forget the third one.
Click.
Yeah. So did you find that in your eosinophil, in your sputum as well?
Yeah.
I didn't see that on your list that much.
Right.
Just looking down.
Right, I did. We've got a raft of data, as you might imagine, so I tried to be selective. The question was, did we see a recapitulation of the Fahy findings in terms of seeing increased expression of CLICA, A, serpin B2, and periostin in our signatures? And so, what we did in terms of the eosinophil signature, they were really housekeeping genes for eosinophils that were really in a very selected way picked for the eosinophil gene signature. And so we weren't actually looking to recapitulate the Fahy data in that setting. They're more housekeeping genes that are specific.
So you didn't look at the...
We didn't look at those. For the eosinophil gene signature, but we certainly have the data. Yeah.
It looks like not even going into personalized medicine, but in terms of gender disparities, asthma seems to be maybe even disproportionately affecting women, and there may be some triggers. So have you ever thought about something in terms of differentiating potential molecular targets, estrogen, progesterone receptors?
Yeah, that's very interesting. The question is, there are some subsets, some clusters of asthmatics that seem to be dominated by women, and have we looked at possible association between estrogen, progesterone genes and asthma for designing new targets. And what I'd say, at least in our area, that wasn't a particular interest to develop a niche drug for perhaps that cluster 3 that we spoke about. I know there were previous talks here, and one recently on obesity and asthma. And there, I think Stephanie Shore presented some very interesting data discussing IL-17 and its expression in the obese asthmatic mice. Whether or not that would translate or whether or not that would be an interesting target for that patient population, I really don't know. But I thought that was an intriguing observation.
Can you comment on any interest in pharma in doing viral challenges and looking at biomarkers? And that's an obvious go-to, but you don't see that much published, at least more you see it in academic literature, but not in pharma.
Sure. So the question was, is there any interest in doing viral challenge? And what I'll tell you is that Merck has been part of the UBioPred network that many of you might know about. It's a consortia of asthma experts, academic sites, and pharma. And as part of that collaboration, actually, we have sponsored a viral challenge study with, I think it's with Ian Pavord and with a number of asthma experts. Chris Breilin's involved. So hopefully the results of that viral challenge will be seen soon. And we plan to do a lot of the same kind of molecular profiling work in that study.
Marcy, can I ask you a little bit of a way out question? Maybe someone in the audience can help me formulate it. It's comparing and contrasting asthma with rheumatoid arthritis and the success of novel biologic therapies in rheumatoid arthritis that we've seen compared to the lack of success in asthma. And I don't know that we're measuring biomarkers in rheumatoid arthritis, that we have utility in the targets for TNF-alpha versus whatever it is, IL-2 or something like that. We just try one, try the other, and the next in rheumatoid disease. Are there similarities and differences that are useful lessons in drug development? I'm sorry if it's a crazy question.
No, I think it's a really interesting question, as all your questions are, Chris. It's an interesting question, and one of the things that immediately comes to mind is, although there's always heterogeneity in a population, in a given disease diagnosis, what's been fascinating about rheumatoid arthritis in particular is that it's very responsive to TNFs. And pretty much, it's very obviously been transformative in terms of its therapeutic effect in rheumatoid arthritis. And so I think maybe you're thinking, biologics really have had impact there, and it may be because there's more uniformity in that disease than there certainly has been in the heterogeneous mix of asthma. I don't know, Elliot, you might have a thought about that.
It's hard to know. I think there is heterogeneity, and certainly in the severe asthma group, much more than in some of the others.
But all immunologic diseases are very tough diseases to try to develop new therapies for, and they all have their unique challenges.
So make a prediction. 10 years from now, will we find the new TNF-alpha blocker of asthma? We hit the nail on the head. Or will we treat our patients with a soup of different molecular targets that together modify severe asthma? Or will we have sub-silos that we will, each nail will be different for different patients? I don't.
Boy, if I had that answer, Chris. Certainly not in 10 years, because as you can see, drug development takes at least 10 years. So it's hard to know. I think really advances in understanding the underlying etiology of asthma are going to be made in institutions like this. And from there, hopefully, we can then develop new drugs. But that's on your shoulders. And then we'll try to apply that knowledge to develop a therapy.
Good. All right, well, I want to thank you again very much for a great presentation.